So all you have to do is listen into everything that I'm going to be saying, then watch the video, make sure you write down all the notes once more in your biology book. And like I always recommend, watch this video at least 3 times. One, to help you write the notes, two, to help you understand, then the third time to help you remember the information adequately. So I have to be somewhere at 14 hours. So you would have to forgive me, but it's an introductory lecture to biology. We're just talking about cells, structure of an animal cell, structure of a plant cell, what exactly is the cell theory by definition. This is a very simple topic, but it always comes in the multiple choice section. Also in paper 2, they can bring you some form of diagram and they say the label the organelle labeled A, B, C, D, etc. And then you state some of its functions, or what are the differences between a plant cell and an animal cell. So this topic is very easy, meaning you shouldn't fail any question under the cell theory. Are you ready? Can I start? All right, all right. Please follow me very, very carefully and watch the videos once more.
Okay.
So this is what we're going to be talking about, because for my lectures, I also give a little bit of a historical background, because for science to make sense, you have to know where everything you're learning is coming from. So I'm going to give you a bit of the historical background. Then we're going to talk about what exactly the cell theory is. You have to be able to understand the cell theory, not just saying, oh, a cell is a basic building block of life and just end there. No. For us in this lecture, we're going to actually go beyond and actually list 3, they call them postulates or ideas. So in science, when you hear something called a postulate, it's an idea or a theory. So we're going to talk about the cell theory. Then we're going to look at the basic structure of a cell. The basic structure of a cell is something known as protoplasm. So protoplasm is found in all types of cells, be it a plant cell or an animal cell. And we're going to look at the individual detailed structure of an animal cell and the individual detailed structure of a plant cell as seen under a light microscope. All right, so let's talk a little bit about the history. So one of the people who pioneered um, cell theory development is a scientist known as Robert Hooke. Robert Hooke. So Robert Hooke was one of the first people who actually discovered cells. He used to play around with a lot of microscopic lenses, as seen here. So he actually developed one of the first microscopes that we can have. You can see that it just had a very simple lens right there and a simple bulb that acted as a light source. So what he did is that he went to a bark of a tree and actually cut a small piece, made a little bit of sections, placed the sample right there on the specimen slide, and what he could see is this structure right here. He actually saw these tiny, minute box-like structures, these tiny individual block-like structures. And because they resembled cells, like, prison cells, where you have sort of cages and little boxes, he called these little structures that he observed as cells. So that's what Robert Hooke did and discovered. But it was later this man here, this Anton van Leeuwenhoek, who actually read Robert Hooke's work, and using Robert Hooke's work, he was able to modify the microscope that he was using, and he developed a better microscope from the one that Robert Hooke was using. So his microscope was way better. Actually, Antoine van Leeuwenhoek is actually considered the father of cell biology. So we owe our study of cells and cell biology. No, it's all right. We owe our foundation of cellular biology to Robert Hooke, because he was actually one of the first people to identify these cells under the microscope. You can see that these types of cells are different types of cell specializations in animals, with the exception of this one. This is a bacteria cell. So he was able to observe different types of white blood cells. When you get your blood, you put it, you get a drop of blood, you put it on a glass slide there, and then you view it on a microscope. You are going to get these types of cells. So he was able to observe red blood cells, different types of white blood cells. So these are the different types of white blood cells. I'll tell you what these types of white blood cells are, but this is an eosinophil, this is a monocyte, this is a basophil, and this is a monocyte. But you don't have to know this. At grade 12 level, you just have to know that an example of cell specialization in animal cells are your different types of white blood cells. This is a reproductive cell. This is a sperm cell. These are non-animal cells. So this is an yeast cell here, and this is a bacterial cell. So using the microscope that he was able to develop, Antoine van Leeuwenhoek was able to demonstrate these cellular structures. Another interesting thing that you have to know about Antoine van Leeuwenhoek is that he was actually not so rich. He came from a very poor background. But because of hard work and perseverance, he was able to make a lot of considerable contributions to the world of science, and actually become a very, very successful human being. So what is the cell theory? So the first thing that you have to understand is that all organisms are made up of cells. All organisms are made up of cells. When we're talking about organisms, all organisms are made up of cells, whether you are unicellular. Uni means one. So if you are why am I combining uni and multicellular? Whether you are unicellular or multicellular, you're going to be made up of cells. So all living organisms are made up of cells. Then a cell is the basic unit of structure and function. So remember, in the introductory video for biology, I told you biology, we're looking at structure and we're also looking at function. So these cells, in the future, we're going to realize that different types of cells can come together like so. So different types of cells can come together to form a tissue. So imagine these, the different types of cells that make up this tissue known as the pancreas. The pancreas, you've seen that. So this is a pancreas. The pancreas is made up of a lot of cells. And the pancreas has got two types of functions. The pancreas has an endocrine function, and it also has a digestive function. So let's talk about the endocrine function of the pancreas. So in terms of the endocrine function, the pancreas has got beta cells, and these beta cells release insulin. And every time you eat a meal that's high in sugar, the insulin is going to uh, um, reduce your blood sugar level. So the insulin is going to facilitate the uptake of glucose into your muscle cells. So that's a function. So have you seen the cell is a basic unit of structure? So it's the cells have given this pancreas its structure, But also these same cells, they are performing a particular function. They are releasing a hormone known as insulin that controls your blood sugar levels. So that controls your blood sugar levels. So this is what I mean when I say a cell is a basic unit of structure and function. So remember, I told you here we have to sort of really understand what we're talking about. We just can't say a cell is a basic unit block of life. If you want to get full marks, you have to tell the examiner that the cell is not just a basic unit of structure. You have to tell the examiner that the cell is also a basic unit of function, because these cells have got secretory functions. They produce certain substances that are needed by the body. The last part is that all cells come from existing cells. So all cells come from existing cells. So every cell has to have an origin. So this here, when we talk about this last part here, we're talking about, I have to change my tip. We are talking about DNA, uh, rep, DNA. No, we're talking about cell division. We're talking about cell division. We are actually going to do genetics at the end, and we are also going to do cell division. Although questions don't come as such under cell division, but you have to know it, because if you want to understand genetics and genes and the units of inheritance, cell division has to take place. So when we say all cells come from pre-existing cells, we're talking about this process here, this process of cell division, where you have got one cell, and then this one cell is going to divide into two cells. Then so this is known as the parent cell, and these are the two daughter cells. These are the two daughter cells. Then these same two daughter cells are going to divide again to make more daughter cells. So that will divide like that, and that one will also divide like that. That one will also divide into two. This one will also divide into two. That one will also divide into two. That one will also divide into two, and so on. So this is the process of cell division. That's what I mean when I say all cells come from pre-existing cells. Then in the future, we're going to realize that this cell division is made up of two types of cell division. The division for the rest of your body, known as mitosis, and the division of your reproductive cells, known as meiosis. So we're going to look at mitosis and meiosis and Mendelian genetics in the future. So the cell theory, the first one, all organisms are made up of cells. The second one, a cell is the basic unit of structure and function within organisms. The third thing, all cells come from pre-existing cells. This is the only way you're going to love biology. If you don't understand biology, then you just be, if you don't understand, you just be saying, biology na eve ina manoti mbili. I can't understand this. So you have to understand the structure and also the function. So in this universe, not universe, on planet Earth, we have got two different types of cells. We have got prokaryotic cells, because we have got bacteria, things like E. coli, salmonella, those things that make us sick. These types of cells are known as prokaryotic cells. Prokaryotic cells. Prokaryotic meaning they don't have a true nucleus, meaning or they don't have membrane bound organelles. So a bacterial cell is going to look like this. And then instead of having a nucleus, the nucleus, we're going to look at the function of the nucleus in this very lecture. The nucleus stores DNA, but its nucleus it's just floating inside the cytoplasm. It's just floating. No wonder it's not called a nucleus, but it's known as a nucleoid. So these cells don't have a nucleus, a true nucleus, and they also don't have membrane-bound organelles. But when we're talking about a eukaryotic cell, a eukaryotic cell is going to have a true nucleus. So let's say maybe this is an animal cell. It will have a nucleus and membrane-bound organelles. So you're going to find a nucleus there. The nucleus is going to have a nuclear envelope. That's the nuclear-bound membrane. So that will be the nucleus with a nucleolus there, and the rest of the structure is going to be the nucleus. So a very true nucleus and membrane-bound organelle. So you might find mitochondria there. You might find some rough endoplasmic reticulum with some ribosomes there. That's a eukaryotic cell. It has a nucleus, and nuclear-bound organelles. So those organelles mean small organs. So little small organs like mitochondria, endoplasmic reticulum, ribosomes, peroxisomes. We're going to look at those given functions. All these organelles are nuclear-bound. So those are the two types of cells that we have. We've got prokaryotic cells and eukaryotic cells. So I've already given you a structure of a prokaryotic cell. So this is a bacteria cell. You've seen this DNA here. There's no nucleus. The DNA is just floating inside the cytoplasm. Later on, we're going to find out that DNA is actually the molecule of inheritance. This is where everything about yourself is stored, from your eye color to your skin color, hair color, your gender, your personality, everything about you can be found in this DNA. This is the basic unit of inheritance from one generation of offspring to another. So in the future, if you have kids, you're going to pass half of your DNA to your kids. Practically, I've got 46 chromosomes. Yeah, we're going to understand this when we talk about inheritance. So when you have kids, if you are planning on having children, you're going to pass 33 of your chromosomes of your DNA to your child. That's why somehow we somehow look like both of our parents, because they gave us half of their DNA. Later on, we're going to talk about all this. All right, so now if a question comes saying, what are some of the features that are common? What are some of the features that are common to both a plant cell and an animal cell? So whether you are a plant cell, whether you're an animal cell, you're going to have three basic structures. And those three basic structures are grouped together into one term known as protoplasm. Protoplasm. And those three basic structures consist of the nucleus, the nucleus, the cytoplasm, and the cell membrane. So whether you're an animal cell, whether you're a plant cell, you need a nucleus, because that's where your instruction center is. This is the brain of the cell. It's the one that tells the cell to do everything. You need a medium where the reactions are going to take place. Where are you going to make energy, respiration? Where are you going to get rid of your waste? How are you going to transport materials from the outside of the cell to the inside, or from the inside to the outside? You're going to need a medium. So that medium is the cytoplasm. Also, you're going to need some form of protection from the outside environment. So you're going to need a cell membrane. So if the question comes and says, what are some of the similarities between a plant cell and an animal cell, you're going to tell them you need a cell membrane, you're going to need a nucleus, and you're also going to need cytoplasm. And all these three structures, this diagram, yeah, all these three structures are grouped together into one structure known as protoplasm. So let's now dive in and talk about the detailed structure of a plant cell and also the detailed structure of an animal cell. Is there a way I can zoom this diagram so that you see it properly? Because I want to explain. Anyway, I'll be drawing the structures here. So if you get some form of cells, so maybe I get a swab. Nowadays, everyone knows a swab because people test for corona. So I get a swab, I swab your cheek cells and I collect those cheek, they are known as epithelial cells. So I get your cheek epithelial cells, I put them on a glass slide, and then I examine them on a microscope. So I examine them on a microscope. This microscope is known as a light microscope or a compound microscope. So I put it there. The microscope is going to have some form of lenses known as objective lenses. And here is where this is where the adjusting knob is. I focus it and then I look at this same structure. So this is what you see when you look at cells through a light microscope, also known as a compound microscope. There are other types of microscopes like scanning electron microscopes, transmission electron microscopes. Those are electron microscopes. But at our level, we just look at the compound microscope or the light microscope. We're going to see these same structures. So let's talk about some of these structures that are important one by one. So we're going to start off with the nucleus. Right here, the nucleus. So the nucleus there contains DNA. So what is the function of the nucleus? The nucleus contains DNA. So that's the nucleus there. Do you see the nucleus structure there? This is the nucleus. This nucleus here contains DNA. DNA stands for deoxyribonucleic acid. So it contains DNA. DNA, so you say it contains DNA. So functions of the nucleus contains DNA. DNA is the instructions of the cell. So instructions of the cell. Instructions of the cell. So DNA is what instructs the cell to do literally everything. DNA is our unit of inheritance. So let's say, for example, the DNA is it's a very cool molecule. So let's say you cut yourself. You were maybe cutting vegetables with some form of sharp knife, and then you cut yourself. Here there's a wound. There's a wound. This wound here, before you know it, a scar is going to be formed. A scar is going to be formed. So every time there's a wound, your cells detect that, here we're losing blood, here we're losing blood. So what should we do? Let's instruct these tumor structures known as platelets to clamp on the wound. So that we stop the bleeding. Then over time, what your DNA is going to do, it's going to start making proteins known as fibrinogen. Then this fibrinogen is going to be interlocked on that wound. So fibrinogen will get converted to fibrin, which is a very hard protein, and over time your wound is going to be healed. So all these instructions in a cell as a direct product of your DNA or the instructions within the cell DNA. So nucleus, what's the function? The nucleus contains DNA, deoxyribonucleic acid, which are the instructions of the cell. Are we okay?
Yes.
All right. So let's look at our second structure. I labeled the wrong diagram. There are two structures in there. So there's this, the nucleus is this. Then inside the nucleus, there's like a structure there, which is the nucleolus. So let's look at the function of the nucleolus, which is just like a small structure inside the nucleus, this structure here. So this structure here, the nucleolus, produces something known as um, ribosomal, it produces RNA. So this is where RNA is made. What's the function of the nucleolus? RNA is made in the nucleolus. So RNA, so the interesting part is that DNA is found in the nucleus, and it's very, very important. Because DNA is very, very important, it should never leave the nucleus. So the cell is actually very intelligent. It's going to make something known as RNA. So RNA is ribonucleic acid. It's this RNA that's going to enter the nucleus, copy the DNA, get the information that's found in the DNA, and then it's going to go outside the cell to make the protein, to make the protein or the enzyme or any other type of material that the cell might need. So the DNA is found in the nucleus. It's very, very important. It shouldn't be harmed. So the nucleolus is going to release something known as RNA. It's going to make RNA, or it's going to make RNA. Then this RNA is going to go outside the cell to start making all these other tumor structures. The other function of the nucleolus is that this is where ribosomes are made. So these tumor structures known as ribosomes, they are made in the nucleolus. Actually, yeah, this is a better way in which you can remember this, because RNA might be a little bit A level. So let me just say, because you know what ribosomes are, you've heard of rough endoplasmic reticulum. Rough endoplasmic reticulum contains ribosomes. So these tumor ribosomes, what's the function of the nucleolus? It makes ribosomes. So the nucleus contains DNA, it's the instructions of the cell. Then the nucleolus here makes ribosomes. We're going to talk about the structure of ribosomes, I think very soon, right about now. So what this nucleus does here, so I've got the nucleus. Just outside the nucleus, you've got a weird flat-like structure. This weird flat-like structure here is known as an endoplasmic reticulum. And this endoplasmic reticulum contains these tumor round structures here. These round structures are known as ribosomes. No wonder it's called rough endoplasmic reticulum. So it's called rough endoplasmic reticulum because it has ribosomes attached to the surface. No wonder it's called rough endoplasmic reticulum. So what's going to happen here? I told you that the nucleus has certain instructions. So the nucleus tells the cell, all right, we need to make proteins. So how do we make proteins? We need to make proteins. So make proteins. So the nucleus tells the cell, all right, we need to make proteins. An example of a protein that I gave was fibrinogen, the one that went to heal your skin. Or maybe uh, uh, you the protein that you grow your nails grow by themselves because they have got protein. Maybe melanin for our skin so that we don't get too much sunburnt. So there are a lot of different types of proteins, maybe even insulin or estrogen or testosterone. All those types of proteins, hormones, and enzymes are all made because of the nucleus. So the nucleus instructs the cells, go make proteins. So where are these proteins going to be made? The proteins are going to be made in the structure known as the ribosomes. So the ribosomes are the ones that are going to make these proteins. And these proteins, they have to be modified to perform specific functions. So you're understanding, right? Where are proteins made? The ribosomes. What is the process in which proteins are made? It's known as protein protein synthesis. So protein synthesis is the process in which proteins are made inside the ribosomes. Protein synthesis is the process in which proteins are made inside the ribosomes. So the ribosomes make these proteins, but these proteins are not yet mature. They have to be modified for them to be ready to perform their specific functions. So what structure modifies the proteins? It's the rough endoplasmic reticulum. So do you see the different structure? So it modifies and yeah, it modifies the proteins. So now the proteins have been modified inside the rough endoplasmic reticulum. So you've seen the steps. The nucleus tells the cell, all right, let's make proteins. Where are the proteins going to be made? In the ribosomes attached to the endoplasmic reticulum. After they are made, they are not yet mature, so they have to be modified. Where are they modified? In the smooth endoplasmic reticulum. Then just outside the rough endoplasmic reticulum, you have got structures like this. These structures like this, they are sort of like stacks. They are maybe stacks of 3 or stacks of 4. This structure here is known as the Golgi apparatus. The Golgi apparatus. Sometimes teachers call it Golgi. I feel weird calling it Golgi. I say Golgi apparatus. Here's the sequence. Step 1, the nucleus tells the cell, we need proteins. Step 2, here, your ribosomes has made those proteins. Step 3, your rough endoplasmic reticulum has modified those proteins. They are mature. Now they are ready to be transported. So those proteins are going to be taken to your Golgi apparatus. What the Golgi apparatus is going to do, this is step 4. The Golgi apparatus is going to package those proteins. So it's going to package these proteins in something known as transport vesicles. So imagine that's a transport vesicle, and the green that I've colored in there, that's a protein. So this protein has been packaged into this transport vesicle. And now since it's packaged, it's ready for delivery. So this transport vesicle is now ready for transportation. Is this protein going to the eye to make your eyes brown or green? Is this protein going to your skin so that you should have nice healthy skin? Is it going to your hair? Is it making proteins like insulin, estrogen, testosterone? So all these different types of proteins are packaged and transported into to various parts of the cell via the Golgi apparatus. So that's step 4. So you've seen these structures of the cell, they make sense, no wonder they are arranged close to each other. They are arranged close to each other. The nucleus is going to be close to the rough endoplasmic reticulum. The rough endoplasmic reticulum is going to have ribosomes. The rough endoplasmic reticulum will be very close to the Golgi apparatus. So your nucleus sends the instruction, your ribosomes will make, the rough endoplasmic reticulum will modify, And your Golgi apparatus is going to package and transport that everything that has been made to various sites within the cell or outside the cell to the rest of the body. Have you understood that?
Yes.
You just have to watch the video to really understand these functions. So now you can wonder that, wow, all these processes really need energy. They literally need energy to do this, because nothing's for free. Nothing's for free. You need to supply some form of effort and energy to perform certain functions. So where does the energy come from? The energy to perform all these cellular functions comes from a structure known as the mitochondrion. Mitochondria is the plural. So mitochondrion is one mitochondria, then mitochondria it's many. So the mitochondrion, they say it's the powerhouse of the cell. Why do they say it's the powerhouse of the cell? Because look at all these functions, they need energy. So this is where the energy comes from. So energy production. Energy production comes from the mitochondria. So what does the mitochondria do? Where's my cursor? What does the mitochondria do? It does something known as cellular respiration. Cellular respiration. So cellular respiration is the process in which cells make energy within themselves, within the body for the function of the cell. So what is cellular respiration? At grade 12 level, it's sugar plus oxygen to give us carbon dioxide plus water plus energy in the form of ATP. So this is adenosine triphosphate. So this is the cellular energy that's released as a product of cellular respiration. And this energy is going to be used by the cell to carry out all its functions. So the nucleus instructs, the ribosomes make, the rough endoplasmic reticulum modifies, the Golgi apparatus packages and transports, the mitochondrion provides all the necessary energy that is needed, to carry out all these cellular functions. And where does all these cellular functions take place? Inside the cytoplasm, which is 90% water. The cytoplasm is 90% water, and 10% are all these structures we've talked about: ribosomes, nucleus, rough endoplasmic reticulum, etc. So the cytoplasm, that's the medium. Cytoplasm is the medium in which all cellular reactions take place. Cytoplasm is 90% water, and it's 10% suspended cell organelles. And all these organelles are nuclear bound. So let's talk about where there is a rough endoplasmic reticulum, there is a smooth endoplasmic reticulum. Where there is a rough endoplasmic reticulum, there is a smooth endoplasmic reticulum. So something known as smooth endoplasmic reticulum. So this smooth endoplasmic reticulum, they say it is smooth because there are no ribosomes on its surface. What does this one do? It's detoxification, detoxification of the cell. So this, what it does, because all these cellular reactions here, they release certain waste substances that can be toxic or harmful to the cell. So what the smooth endoplasmic reticulum is going to do, it's going to help to detoxify the cell but also it produces certain lipids and fats, because later on I'm going to tell you that the cell membrane is a lipid bilayer. The cell membrane is made up of lipids. Where do these lipids come from? They come from the smooth endoplasmic reticulum. The last cell organelle I want us to talk about with the animal cell are these structures here known as the lysozymes. So the lysozymes are the maids of the cell. They are the ones that clean up dead cells, debris, waste products. They are going to move around, inspecting, surfacing around, cleaning up the cell. So that's the function of the lysozyme. Oh, there is one more very important structure, the cell membrane. The cell membrane. The cell membrane, extremely important cell structure. So let me draw for you a cell membrane so that I show you how it looks like. If you look at it properly under an electron microscope. Let me draw it a little bit lower because I want to explain certain things. Where's my cursor? I always lose this cursor. So the cell membrane looks like this. It has got phosphate heads, and it also has lipid tails. So these two ones that I'm drawing there are lipid tails or fat tails. Then these are the phosphate tails. Phosphate chemistry, there's an element there known as a phosphate. So these are the phosphate tails. And these phosphate tails have got certain charges. They are negatively charged. They are negatively charged phosphate tails like that. So here there's a negative charge, then this is the inside of the cell, where you have your organelles, your nucleus, and your cytoplasm, everything else. So here what's going to happen here is that if you have got certain ions, certain charged ions, so maybe this is a substance here, an ion, that's negatively charged. Negative plus negative, they repel. So this won't pass through. Or if you have something that is big, something that's very big, it won't be able to pass through because it's too big to squeeze through. It's too big to sort of squeeze through the cell membrane so that it can enter. So it won't pass. But if you have something that is neutral, for example, water, and certain gases like oxygen and carbon dioxide, which is a waste product, they'll be able to pass through and leave the cell. But these other ions that are negatively charged and these other things that are very big, they won't be able to pass through the cell. In this way, the cell membrane is selectively permeable. In that it can only allow certain important things to pass inside and outside the cell. Why important things that are neutral? Water is very important by the cell, so water has to be able to get into the cell, it has to be able to leave the cell. Oxygen is needed for respiration, for your mitochondria to produce that energy. Carbon dioxide it's a waste product, it's toxic. If it accumulates too much into a cell, the cell can die, so it has to leave the cell. So these are very important substances that can pass inside and out of the cell. But certain ions, big molecules that might be harmful to the cell, won't be able to pass through the cell. Therefore, this is what is called selectively permeable. So when they say what is the function of the cell membrane, it is selectively permeable. It can only allow important things that are needed by the cell, like water, carbon dioxide, and oxygen. And it can only allow important things that are needed by the cell. Have you understood that?
Yes.
All right, thank you very much. So this is the animal cell. And by the way, animal cells also have vacuoles. They are just not big, they're just very small, because sometimes the animal cell can be in a very salty environment. And when it's in a very salty environment, the animal cell can lose water via osmosis. We're going to talk about diffusion and osmosis in the next class. When these cells lose water via osmosis, they can shrink. So to prevent them from dying, they can also store some of the water and electrolytes into their small vacuoles. So a vacuole is also found in an animal cell. If a question comes, what are the differences between a plant cell and an animal cell? And then you say a plant cell has a vacuole while an animal cell does not have a vacuole, you're going to get it wrong. All right, so let's now look at different types of cells. So I've explained everything. You have to write down these notes. I have explained it in one diagram, but all these notes, you're going to find them. And I want you to write each and everything here. Cytoplasm, structure of cytoplasm, the nucleus, write everything, draw what you can. If the diagram is too complicated, it's fine, but draw what you can. So ribosomes, protein synthesis, uh, use the video to understand. Write down the notes, but use the video to understand. Some of them might be a little bit complex and A level, but use the video to understand. Rough endoplasmic reticulum, where it has got ribosomes, smooth endoplasmic reticulum, no ribosomes, function of rough endoplasmic reticulum there. Those are the functions. So please use this video, because some of these notes might be a little bit advanced, but write them either way. You're a student. The mitochondria there, powerhouse of the cell, ATP, lysozymes. These are the ones that clean the cell. They have these digestive functions where they sort of clear out the debris of the cell. Peroxisomes. So peroxisomes, what they do here, cellular respiration, all those other products produce this substance here, H2O2. It's known as hydrogen peroxide. So have you heard that, okay, vitamin A, vitamin C, vitamin E, they are antioxidants?
Yes.
So this here is an oxidant. It's an oxidant here. It damages the cell. It damages cells. So what's going to happen is that certain organelles known as peroxisomes, they are going to convert this hydrogen peroxide to water and oxygen, which are not harmful at all. So that's what peroxisomes do. They convert hydrogen peroxide into water and oxygen. Actually, even certain vitamins like vitamin A, vitamin C, vitamin E, they are also antioxidants. They clear out harmful, they call them free radicals. They clear out harmful radicals like hydrogen peroxide. So the only structure of the plant cell that I'm going to talk about is the cell wall and the chloroplast. The rest are the same. So these are the two structures that are only found in, these are the only two structures that are only found in plant cells. Then I also talk about the vacuole. But remember, vacuole is also found in animal cells also, but you have to know the function of the vacuole in plant cells. So the cell wall contains cellulose. So here you have to know that cellulose cell wall, the cell wall contains cellulose. Do not forget this. It contains cellulose and it also gives it a support, support, and also protection to the cell. So it is a cellulose cell wall. It gives the cell the structure, and it also gives the cell the support. Then what do these chloroplasts do? This is where photosynthesis takes place. We're going to look at photosynthesis very well in detail when we look at plants in general. So what does photosynthesis do? Um, Sunlight. What's the formula of photosynthesis? In the presence of sunlight and oxygen. Wait. Oxygen, water, water plus oxygen in the presence of sunlight and chlorophyll. And chlorophyll, oh no, this, it's carbon dioxide. Yeah. So water plus carbon dioxide in the presence of sunlight and oxygen to give you oxygen plus glucose. So this is the formula for photosynthesis. So this reaction here, water plus carbon dioxide in the presence of sunlight and chlorophyll to give you oxygen and glucose, takes place in the chloroplasts. We're going to talk about this process in detail when we look at photosynthesis and experiments for photosynthesis. You put a plant in the dark, what's going to happen? Is it going to photosynthesize? How do you prove that photosynthesis has taken place? We're going to look at this in detail. Then the vacuum is for uh, water water plus electrolyte balance in the cell. So because plants need water for them to carry out photosynthesis, sometimes plants can take up too much water and become turgid. Sometimes they can be drought and plants might start losing water. So plants need a reservoir of water. So to keep that water and electrolyte balance in place, plants have this structure known as a vacuole that keeps water and electrolytes. So vacuole keeps water and electrolyte. So it's for a water and electrolyte balance within the plant cell because primarily plants rely on water for photosynthesis. All right, there are certain notes. There are cell wall, chloroplasts. There is a structure known as plastids, which are not so, this is A level, they are not so important, but they are storage granules. All right, so that's the end of our first lecture for biology. In our next lecture, we're going to talk about different types of cell organization and types of cell specializations in plant and animal cells. Then we're going to dive right into human biology, where we're going to start looking at the organ systems. We'll start from the circulatory system, then the respiratory system, digestive system, endocrine system, reproductive system. We're going to make sure we know human biology very well, including DNA and Mendelian genetics. After we know human biology very well, we're going to transition to plants. We're going to talk about transport system in plants, reproduction in plants, what are monocotyledonous plants and dicotyledonous plants, what is photosynthesis, etc. So I'm going to separate the biology into human aspects, then we'll do the plant aspect at the end. So this is the end of the lecture. I will send you the notes, but first I have to rush into school. I'll send in the notes and I will see you tomorrow. Bye.
Okay.
All right.